everyone. My name is Astrid, and thanks in advance, everyone, to joining this presentation and listening. And also thank you, LabRoots, for the opportunity to speak here. So as I said, my name is Astrid. I'm the founding manager and managing director of Upcyte Technology. We provide expanded liver cells for research and development, and I'm going to talk today about the generation of expanded primary liver cells. So as you can see on the slides, the content is going to be the solution of Upcyte Technology, the technology itself, and generally what we offer, a brief history of the cells and the publications. And I'm going to dive deeper into the actual science of the hepatocytes, the liver sinusoidal endothelial cells, and other cell types we provide for R&D. So what we're trying to offer is we're trying to find the best cellular model on the market. So as you all know, for hepatocytes, for example, on one side you have the cell lines, that's HepG2 or HepRG. They come with a high availability, so they proliferate. You can use them for screening applications. And on the other side, you have the primary hepatocytes, which have a high physiological relevance, but sometimes the tissue is hard to get, or the lot sizes are quite small. So we're aiming for a compromise model, meaning we're trying to offer a cellular model which has, on the one side, high availability, and on the other side, has high physiological relevance. So this is a more holistic approach, so that's suitable for cells that in general do not proliferate in vitro, such as the hepatocytes, or where the tissue is hard to get, or in general where we have low cell yields, or also if you have a patient-derived material where you just get a few 100,000 cells, for example. So how do we do that? Here you see a brief overview. So we either isolate the cells on our own, or we buy them from commercial vendors, or we also do have a network of university partners where we get the primary cells from. So we usually work with adherent cells. So we take the cells, either fresh or cryopreserved. We thaw the cells, we put them into a plate, and we have a genetic library of different genes and gene combinations, which we try out. So we're using a lentiviral transduction, so we're genetically modifying the primary cells. Usually we do that in a 6-well plate, where we try out different genes and gene combinations, as I said, but also different media supplements, and we transduce the cells and then we wait. For the hepatocyte, that process of the cells to start to proliferate can take up to several weeks. It's easier for cells, for example, for keratinocyte skin cells that already do proliferate. So at some point, the cells start to proliferate and we can expand the cells in-house. And of course, we want to check if they are still liver cells. So we do a quality control. We check for the prototypical liver markers. I come to that later in the presentation. And it's really important to mention that these cells are not fully immortalized, so we're not generating a cell line. It's a little bit dependent on the donor as well as the cell types, how far the cells proliferate. So for example, for keratinocytes, the skin cells, as I said, they do proliferate up to 60 or 70 population doublings. For the liver cells, who are focused in that presentation, they do proliferate up to 30 or 35 population doublings. So as soon as we check if we actually still have liver cells, we do a production, we further expand them, and at the end we're selling the cells ready to use. I come to that production scheme again in 2 slides. So as an example here, you have hepatocytes. There are 2 different donors. So you see one donor at the bottom and one donor on the top. So we see the cells, as you can see on the left side, subconfluent, meaning they still have space on the plate to proliferate. So the left side is still showing the primary human hepatocytes directly after seeding. Then we do the genetic modification. We take the lentivirals, put it in the supernatant on the cells, and transduce the cells. And after some days or weeks, you can see little islands of cells growing out of the well, or basically out of the cells. And then we do a first passage of the cells. So that's the crucial step as when they survive the first passage, you can you can treat the cells as you would treat normal cell lines. On the right side, you see as soon as the cells actually hit confluence, they are contact inhibited and they start to differentiate. So we do have a weekend-friendly protocol, so the cells are slightly differently treated than primary hepatocytes. You thaw the cells, you give them a recovery phase, and then you can start using them for your experiments. On the bottom, you see a different donor. The primary cells directly after seeding look really nice and fit, but after the lentiviral transduction, you can see that there has been a contamination with other cells. So the primary hepatocyte, or the island of primary hepatocytes, are surrounded by fibroblasts. And if that's basically to show if the donor is not pure, it makes our life really hard, because we upcyte everything, all the cells which are in the well, and we have further purification steps where we try to really just isolate the hepatocytes. But it's just in general to show the purer the donor, the better for us. If you're experienced with the different donors, so we have different donors available, you can also see that the morphology of the differentiated cells is different, and you can really see which donor you are working with if you're experienced with the different donors. So as I said before, I said that I'm going to explain the production scheme again. So as I said, we start with the upcyting. We generally we can start with a few 100,000 cells, but it's preferably to have more, like 5 or 10 millions, or basically a vial of primary hepatocytes. We upcyte the cells, we expand them, we freeze a backup in between, and we try to expand them to a master cell bank. And we're already checking initially from typical hepatic markers to prove that these cells are still hepatocytes. We freeze down the master cell bank, which is typically 10 vials consisting out of around 10 million cells, with a population doubling of around 15. And from there, we can expand further to a working cell bank, which we then sell ready to use. So as I said, the cells are not fully immortalized. We expand them in-house and provide them cryopreserved, ready to use, with, for example, our protocols, which you then can follow for the different experiments. With that production, we can generate up to 3,000 vials from the same donor. So that is way more than you can achieve with just a typical primary hepatocyte isolation. So the advantages of the technology are that, as I said, you can create up to 3,000 vials from a single donor, which makes them suitable, for example, for screening applications. They do have the primary cell features, so we always start with healthy human cells, so they're not generated from a cancer, or they're not a cancer cell line. They're karyotypically stable, they have a physiological relevant profile, and usually the cell type specific phenotype. They're easy and safe to handle, so if you experience in general with primary hepatocytes, it should not be a problem. From the handling side, they're more comparable to a cell line. We have protocols for use, which we provide. We have detailed information on each donor, and also they come along with optimized media. We do, for the hepatocytes and the other liver cells, we have different donors available. And we also do custom upcyting of primary cells. I'll come to that in a second as well. And you can basically use the cells for everything you use the primary cells for. So they're flexible in use. You can do, for example, 2D or 3D cultures, or co-culture with other cell types. One highlight to mention is here that they actually survive in cultures and have stable metabolism up to 4 weeks. That is an advantage over the primary cells as well, because usually the primary hepatocytes dedifferentiate quite fast in culture. So as a summary, as I said, what we offer, on one side, we have the ready-to-use cells. They come in 5 million cells per cryovial. We also do have a license model if you want to expand your own, to come along with cell culture media, which is specially designed for our cells, but you can also use primary cells. So we do have a lot of customers who are actually using primary hepatocytes, but using our media. For the upcyte service, that comes in handy when you, for example, have a specific cell type or you have a favorite donor. Usually the pharma companies have a favorite donor when it comes to hepatocytes, or when you have patient-derived disease material. That's a flexible, milestone-dependent offer. We can do a basic characterization of the cells. We can do like an extensively characterization of the cells. We can expand the cells and do further customization. For example, we had a really nice project last year where we had a knockout mouse and we isolated the hepatocytes from that mouse and upcyted the cells, and they still had that knockout after upcyting, so the scientists can continue working with the liver cells of that mouse without having to kill a mouse every second week. So if you go into PubMed, we have quite some publications available. That's specifically for the upcyte hepatocytes in regard to toxicity testing, but also to transporters or, for example, metabolism. So the list is available on the homepage, but also, just go into PubMed and type upcyte in there. Now I come to the scientific part of the presentation. So for the hepatocytes, as you all know, there are current problems of the hepatocyte model. So on one side, you have the primary hepatocytes. They might be limited in lot sizes and availability in general. You have huge donor differences. So, and also within the lot, you have a great variability. So it's kind of time consuming to qualify each lot again. And usually the culture is limited to some days. If you have a monoculture, if you do co-cultures, they can survive for longer, but they also de-differentiate in culture. On the other side, you have the tumor cells, for example, HepRG, HepG2, which are great cell models. It's also always dependent on what you're looking for and what your output of the experiments are. But here, you don't have a donor choice. They are cancer-derived, and sometimes, for example, for HepRG, it's a mixture of two cell types, and you also need a high concentration of DMSO in the media to actually differentiate the cells. You also have iPS-derived cells. I think, in my opinion, that works quite well for other cell types, but it does not work for liver-derived hepatocytes or iPS-derived hepatocytes. The differentiation state is still in an embryonic stage. So, in my opinion, this doesn't work for hepatocytes. When you look at our cells, they are primary-like phenotype. They're metabolically competent. They have stable metabolism for weeks in culture. We have data showing that they are proliferating. You can also transfect them. We have different donors available. We also have one disease model, which is an enzyme defect, which is not quite known, but the Wikipedia entry to hyperoxaluria is great. And you can basically use them for everything. You use the primary cells or the cell lines, for example, cytotoxicity, genotoxicity, induction clearance, and further applications. So how do we know that our cells are still hepatocytes after we upcyted them? So what we do in-house is, first of all, check for the morphology. So the difference in morphology from hepatocytes to, for example, a fibroblast is quite clear. So as soon as they hit confluence, they have the typical cobblestone morphology. And what we do test in-house is typical markers as, for example, cytokeratin 1818, as well as human cereroalbumin. And if you let the cells stay in the culture for longer than 5 days, they form biocaniculae. And that's all data which is published, and to basically show that our hepatocytes are still liver-derived. What we also do in-house is we check for the enzyme activities. So in-house, we do 1A2, 2B9, 2C9, and 3A4. As I said in the beginning, it's a compromised model, so the activities are not as great as when you have fresh primary hepatocytes, but they're on a similar level than, for example, if you compare them to HepRG. So we're quite good with, for example, CYP3A4. We're not, or the cells don't have a great 1A2 activity, but we always working on improving the upcyte process, and we're quite confident to show or increase the activities, for example, for 1A2 within the next year. For the enzyme themselves, so we can induce the activities with prototypical inducers, for example, rifampicin, but we can also inhibit with prototypical inhibitors. The phase 2 activities, which you can see on the bottom right, they're similar to primary hepatocytes, so they're really well retained after the upcyting process. If you look at the transporters, that's actually a published paper from Boehringer Ingelheim from 2018. On one side, they checked for the sinusoidal transporters as well for the canalicular transporters, and the experiments were divided in 3 phases. One was the mRNA expression, the other one protein abundance, and then actually the active uptake of compounds. So on the sinusoidal side, it does not look great. So the transporters are, if you compare them to primary hepatocytes, are quite low. And for example, ORTB1, B1, and B3 are very low. So they also show a lack of active uptake through those transporters, but the active uptake, for example, for NCTP and 2B1 and OCT1 worked quite well. On the canalicular side, the transporters are quite well preserved, so they're comparable to the primary hepatocytes. One note here is that they actually just tested 3 of the 8 donors which are currently available, and we don't really have data of the transporters before upcyting, so it could be that they were already quite low on the ORTP side, for example. And also, they did some media evaluation. If you take out dexamethasone, for example, from the media, it improves the ORTB1 performance. So there's still a lot to discover and a lot to play around with the media composition, for example, or the culture conditions in general. But that applies to the transporters as well as the metabolism part. When you look at toxicity testing, I just put a summary slide together here because we have quite a few publications. So we have comparison from the upcyte hepatocytes to primary human hepatocytes, but also HepG2 for different endpoints as ATP, MTS, also 24 hours dosing, as well as 1 week dosing. And one paper I like to highlight is the first one from 2016. It's a paper about long-term toxicity testing. So as I said, one advantage of those cells is that you can actually culture them quite long, and that opens possibilities which you don't have with primary cells. So you can really do repeated dose toxicity testing up to 3 weeks, which you can't definitely do with the primary cells. So in summary, as I said, for toxicity testing, we have quite a lot of data available. Also, for example, cholestasis, apoptosis, or steatosis testing. And since they do proliferate, you can also use them in gene tox, for example, for the micronuclei assay. You can also transfect the cells. We have tested a lot of transfection reagents in-house. For example, Virome-Red, which we think is a great one. We transduce the cells with GFP and so on. You can use them for metabolism, what I just presented, for long-term clearance, for example, since they are stable for so long in culture, but also in 3D culture. So you can co-culture them, for example, with the liver sinusoidal endothelial cells or Kupffer cells. You can use them in printing devices, spheroid cultures, for example, the lower attachment plates, also fluidic liver chip systems, and put them on any different scaffolds or matrices. Other experiments or applications are, for example, insulin-dependent metabolic activity assay, but also you can use them for viral infection. For example, we have data which is published for HCV. All over, it's, as I said from the beginning, it's a compromised model. So from the functional side, they're quite good. The main advantages, just to highlight them again, are the handling and also the lot sizes and also the long-term culture. So as an alternative to primary human hepatocytes, which is currently the gold standard, it depends a little bit on the application you might want to use them for. I don't see it as a replacement for primary hepatocytes. I rather see it as a complementary model to primary hepatocytes, and a way better physiological model than, for example, HepG2. The second part of my presentation is actually about the liver sinusoidal endothelial cells. So a lot of people do work with hepatocytes, but not a lot of people work with the other cell types of the liver. So the L6 are basically part of the non-parenchymal cells of the liver, and same for the Kupffer cells or the stellate cells. And it's quite hard to isolate them. So if you have a piece of liver tissue, you generally can isolate millions and millions of hepatocytes, but just a few 100,000 of those. And they do not proliferate as well. So the L6 are lining the blood vessels within the liver in the highly specialized cells. So they are scavenger endothelium within the liver. And to them, we did basically the same as we did to the hepatocytes. So we upcyted them, and they're quite known for three main receptors. So they do have a high uptake within the liver, and they also take up a lot of toxins and transfer them to the hepatocytes. So the most common known liver-specific uptake receptors are the mannose receptor, the scavenger receptor, and CD32B. So as I said, we can generate, we started with a few 100,000 cells of those, and we can generate up to several thousand vials of the same donor as well. And what we did is we characterized them for typical endothelial cell marker as well as typical liver-specific markers. So here you can see the morphology. So the cells are actually quite pretty. The typical endothelial cell markers are, for example, CD31 or tube formation in matrigel. But to really prove that they're liver-derived, we also take a test for the mannose receptor or, for example, the uptake of FITs of albumin, and we stain for LIFE1, which are more liver-specific markers. So there's the research has been limited due to the lack of primary cells. So as I said in the beginning, they're quite hard to isolate. And if you go into PubMed, you will find a lot of publications about hepatocytes, but just a few ones about the L6, because yeah, if you can't really isolate the primary cells, you can't really do a lot of research on them. So if you were in the field of toxicity testing, a lot of people just use the hepatocytes, but kind of leave out the other cell types of the liver, but they're quite important as well. So we're currently writing a publication to actually show that for specific compounds, the L6 and the hepatocytes have different sensitivities. And also when you use them in 3D culture, the toxicity or the toxic response to different compounds is different than when you just use the hepatocytes alone. Also, they're quite important in the immune response of the liver. So, for example, IgG particles are taken up by the CD32 receptors. And also they do play an important role for antigen uptake and antigen presentation within the liver. So there's definitely a lot more to discover. And yeah, we're really hoping that we can convince people who are working with hepatocytes also to look at the other cell types of the liver. What we have done with a customer who's, or let's say a collaboration partner, they have a liver on a chip system. They call Dynamic 42, and as you can see, they have a perfused liver chip system where they have an upper channel and a lower channel, and you can basically seed on one side, you can seed the hepatocytes, and on the other side, you can seed the L6, and they mimic the typical space of this within the liver. And what you can do is also include, for example, blood-derived monocytes, which represent the Kupffer cells, for example, into that chip. And another option would also be perfusing with T cells for the antigen presentation. So there are a lot of possibilities, and there's a lot of research currently going on in regard to co-cultures and 3D cultures, and we're really thrilled to see more. So in summary, what we offer, we have the upcyte hepatocytes I've been talking about. We have the upcyte L6. We have different donors available. We're also working on Kupffer cells, but to admit upcyting cells of the immune system are quite tricky. So we have a PhD student who's also working on monocytes and natural killer cells. We also have available keratinocytes, for example, or microvascular endothelial cells or skeletal muscle cells. And we currently got a grant project to work on enterocytes, cells from the intestines, as well as adipocytes, which are basically the fat tissue. As I said at the very beginning, we not only offer the cells, we also have media which comes along with the cells, which is specially designed for our upcyte cells, but can also be used for primary cells. So if you have any further questions, feel free to write me an email. We are officially distributed by BioIVT worldwide. So either you can send them an email or me an email or call. And thank you for listening. And yeah, have a great day.